presentación del seminario. El seminario es especial porque lo presenta Anita Rich. Anita Rich es investigadora del WSL, que es un instituto de Suiza, que es un instituto federal para la investigación en bosque, paisaje y nieve. Y hemos colaborado con Anita desde hace más de desde el 2012 o 2010, porque participa en la grassland. Anita se doctoró en Suiza en el ETH, que es el Instituto Federal de Investigación. Y es el mismo instituto donde Rolando se doctoró en los 60. Después hay historias entre los compañeros de oficina de Rolando y Martin Schultz, que es del grupo de vida. Hablo en español despacio. Anita entiende español, habla un poquito, está practicando español y ahora puedo presentar en inglés. Hizo su doctorado ahí, pero antes había hecho una maestría en el ETH también y después había hecho una maestría en Michigan. Hizo su doctorado en Suiza y después se volvió a hacer un postdoc a Syracuse, donde se cruzó en algún momento con Martín también en una celebración por Magnópolis. Así que en el 2010 inició su experimento en Dalga, que es donde trabajamos con Enrique, con Laura, en ese experimento que tenemos en Chicas. Y en 2016 nos visitó por primera vez, después de mi acto fallido de decirle, ¿por qué no venís a Argentina? Como alguien invita a alguien a, ¿por qué no pasás mañana? Well, don't you come to Argentina? Yes, I will go. Y vino a Argentina y hicimos un par de publicaciones con eso y ahora está en Buenos Aires con una ayuda de la Universidad de Buenos Aires para investigadores del extranjero. Y ahora nos va a presentar su trabajo de los últimos 10, 20, montón de años de investigación en pastizales del mundo. Anita puede responder preguntas va a responder en inglés, pero si quieren preguntar, pueden preguntar en castellano muy fácil o si no, traducimos. Así que muchas gracias por dar el seminario y thank you very much for your seminar.
Gracias por la introducción y bienvenidos. Pienso que es mejor si hablan inglés para vosotros. Let's switch to English. Yeah, what I would like you to take you through today is a world tour around the world of things that in the last, some work is older, some work is newer, but it's all on grasslands, and to understand what happens if we lose above-ground animals in grassland ecosystem. Not just what happens if they're gone, but what happens to the functioning of the system. What can we afford to lose? I mean, we cannot afford to lose anything, but what happens if animals disappear from the surface of the world? And you may be aware, you've probably all seen this pretty old figure from Rockströmedal, which shows the planetary boundaries that we already overpassed. So what is in red and going over these lines means that the world is already way beyond where we should be. So the biggest thing here, so this is here. This is nitrogen fertilization. So already here we have thrown out way too much nitrogen. But the biggest thing we overpass actually is biodiversity loss, or at least it's a pretty old publication, but 2009, this came out as the biggest problem we have. So this is what I want to get at today. And I mean, you all heard about the defaunation of the last million years. We had several defaulations, the dinosaurs disappeared and other things, but this was not really caused by ourselves. But now we're talking about the sixth mass extinction and the defaunation of the Anthropocene, which is mostly our fault. And we're talking about the trophic downgrading. So we're first shooting the predators, then we're shooting the large animals, and then also we're shooting them by body size. Mostly humans are responsible for the disappearance of like the big including rhinos and everything that existed at one point, and then we're taking out all the smaller things. There's still stuff left, but we're not doing too good to them. And then you may also have heard in the last couple of years the discussion about the disappearance of the insects as well, with all the pollution for the herbicide, insecticides, and also changing the landscape. Even the insects are starting to go. Now I know there's a big debate about this. So some people say, no problem at all, while others say, no, this is a really severe problem we have. So this is what we're facing today. You're all in ecology or agronomy, so you're all aware of that, but I think this is always difficult to talk to the general public. And why do we have to care about biodiversity? And if they see something like this, most people think, oh yeah, they're kind of cute, maybe we shouldn't lose them. Yeah, they understand this part, but when they see something like this, you can okay, this is kind of gross, so why do we worry about hyenas? That's brutal and thing like. And then if we go into something even more problematic for most people, mosquitoes, spiders, they say, well, yeah, okay, if the mosquitoes are gone, good riddance, we don't get stung anymore, so better life. But we all know insects are really important for pollination, they're food for many other things, and also they're important for ecosystem function. So with that, I guess we have a big question. That's what I tried to go at today. What happens if you lose vertebrates and invertebrates, and how does that affect ecosystem processes and functioning? Let's first go into some just definitions, and that you see what I'm talking about. So we have plants, and that get eaten, or these get eaten by large ungulates, for example. That affects the ungulates, that affects the plants. The same happens with soil, carbon, soil nitrogen. The animals affect the soil, the urine and dung goes back, but they also affect the soil through plant soil feedbacks. And then, of course, we also have the whole community in the soil, bacteria, fungi, invertebrates, that's affected also by the large ungulates. Then, of course, we have other herbivores that eat on these plants, small mammals, and all the insects as well. And why ungulates and why rodents? As you see here, there's a lot of bats. These are the rodents, these are the bats, and then others. The individuals as well. There's a lot of, that's the, hard to see. I think that's still the bats. But if we're looking at biomass, that green stuff here, that's the ungulates, and that's actually the rodents. So with the rodents and the ungulates, we're covering a lot of the world's biomass of mammals. There we go. And then, of course, we have a whole trophic food web in the invertebrate system. We have the spiders and the predators that eat the other insects, and of course the deer also, for example, they often ingest the insect by accident. If they eat the grass, they just eat whatever is probably good protein. And then we also have the whole network going on in the soil. And then, of course, in some cases, we have predators or large predators that eat the mammals as well. So why do we worry about the biodiversity loss and ecosystem functioning, and how could it potentially function? This is a theoretical slide, also pretty old, but I think it gives a pretty good impression of things that could happen when we lose biodiversity, but I think we still do not fully understand. So the theory behind this is more or less, it's quite simple. We have three scenarios. This is the first one. So if we start losing biodiversity, at first nothing happens, and then we crash. It's like in a plane, you start losing screws on the wing, 1, 2, doesn't matter, 4, 5, it still doesn't matter. If you lose 200, probably something's going to fall off. That's really important to get you home. The other possibility is this way, so we lose biodiversity and immediate crash. That then probably a jet comes off the plane or two, so it should not happen. And this means here we have redundant species that keep the function, so they can replace each other when one's missing. Here it means that they're all important and we crash. And then, of course, we have the intermediate way. A screw falls off, another one, jet, eventually. Go down too. So I think that's the idea behind that whole thing. And of course, we have this above ground and we have this below ground. And we have redundant species in both, like the brown and the green world, but we don't really understand, I think, to this point, which species are functionally redundant and which not. And then, of course, these two compartments also interact, which we should know what's happening there. But yeah, that's the big question. We get back to that. What I would like to do, take you around the world on different projects I've been in the past and try to answer this question or get at least tackle it down a bit more. Let's start in Switzerland, where I did one large study in the Swiss National Park. We have one national park. It's really small, 72 square kilometers. Really, that's ridiculous small, I think. And we don't have another park. That's complete wilderness. No hunting, no fishing, no camping, no nothing. You can go on a hike, but you're not allowed to leave the trail. If you leave the trail, the rangers will put you back on the trail. And it's completely protected, no management. There's no reduction of animal species. Maybe if it burns, they would put the fire out. It's just that not everything goes up in the air. We have some small nature parks, but they're usually still have management in them, still farming, and they're not really protected. So this is the only one that's EOCN category of myself. We try to get more parks, but the population, the local people always can vote on that. And the local people usually say, no way, we're going to give away our land for no management and that. The interesting thing, the park is now almost 110 years old, and it was established originally with the purpose for research. So see what happens if you abandon an area in its high elevations in the mountains in the southeastern corridor. Pretty rocky too. And it's a dolomite system. Dolomite has a lot of magnesium. If it's taken up by the plant in too high levels, it's toxic. So it's a very low productive system. That's one of the reasons why it became a national park. Not much value in that area productivity-wise. We have a lot of animals in the area, deer, chamois, and the ibex here, that's this guy. Then we have a lot of small rodents like marmots and a lot of insects. When we did the study, there were no large predators in the system. They were all extinct in the last, like the 18th, 19th century. But just 2 weeks ago, we found evidence for the first pack of wolves that came back 100 years after extinction. They ate my insect traps. So the baby wolves playing, we ripped everything out of the ground, and we put the camera on the wolf pack, which is called a couple to at least 4 pups. So this is pretty nice. So hopefully eventually we will be able to also study wolf impact, which is good. So what did we do? We set up an experiment in which we tried to exclude the animals from large to small. What happens also with the downgrading. What we did is we had a big fence here, plot outside where everybody could have access. They went in and grazed. Then we put up here this electric fence, and inside the fence we had the 4 plots. In this one, this means large, medium, small mammals and invertebrates. Then we had a plot open, no fence, just the medium mammals, small mammals and insects. We had an electric fence that keeps the marmots out, so it's not electric, it's a sheep fence, electrified. It doesn't have power at the bottom part, so it can go in. Then we had something like a chicken wire double around this plot so that the small rodents stayed out. And finally here we had a mosquito mesh covered box to keep all the insects out. Looks easy to work. It's a national park. We're not allowed to have any powered tools but a hand drill. So we had to dig out every single of the walls by hand. We had to go 80 centimeters down, and it's pretty rocky. So we had a lot of people trying to get down to the ground and pouncing the rocks by hand. But eventually, it looked beautiful, at least to me. Everything was set up. It worked very well, the design, except when we had snow in summer, then it looked like this, and we had to go back in and restore everything. In the winter time, we had to take everything out because of avalanches, so we had to fold down the fences, take the posts out, and go back in spring to set everything back We had 18 replicates throughout the whole park, so it was a very high-intensive labor project. Luckily, we had 5 years of funding from 2009 through 2013, but still not everything is published. We still have data trying to get out. So, what happened? I split it up into below-ground thing, above-ground things, and ecosystem level, and you will see these icons in the top corner here on the next slide, so you know where we are about in the system. First of all, what blew me away the most was to see who ate what, and as you can see here, this is what the ungulates ate, so 3% of the available biomass, and this number, 26%, that's almost as high as the ungulates, but that was the invertebrates. We were blown away that they eat almost the same amount as the big ones, and you can see some grasshoppers jumping, some cicadas, but not that they would eat that much. But there's a lot of Disanoptera, aphids that suck on the plants and reduce the biomass a lot. So that was for me the most mind-blowing result. And then now let's go through the result, just simplified because you see here, so these guys, marmots and the small rodents didn't really do much, so what I will present is just if you lose all the mammals for this easier presentation. If you look at the plants, what we found that of course when you take the mammal out, biomass increases, competition falls away, and there's more to have. The structure increases mostly because the plants get taller, and but the richness decreased. So we started getting more tall-growing grasses that are competitively strong, that kind of beat out the others. And we kind of changed from a biodiversity-rich grassland with a lot of forbs to a grassland with more grasses. Let's look at what happened to the invertebrates. At the same time, we found an increase in herbivores, of course, more food, more herbivores, more to eat, but also an increase in predators and in omnivores. They all increase because they have more, they get more food, they're really happy. But when you look at richness, the herbivore diversity went down, the predator diversity went down, and also the increase in structure had a negative effect on richness. So if we lose the mammals, we're losing a lot of richness, not just in plants, but also in the next layer. If you look at soil properties, soil temperature went down, soil moisture went up. It kind of makes sense. We're talking about the high elevation system, it's cool in general. You have more vegetation, more shade, so the temperature drops, and evapotranspiration. But whatever else we looked at in the soil carbons are nitrogen, phosphorus, anything, no change at all. It just remained the same after 5 years. If we started looking into composition of bacteria in the soil, same thing, no change at all. Abundance didn't change, richness didn't change, composition remained the same, pretty alert, which was astonishing because other things happened quite dramatically. If you look at the nematodes, we had a huge increase in abundance, and again, as we saw before, a huge decrease in richness. So again, taking the mammal out for richness is not a good thing, but we have more, but more of the same. Then what we did as a next thing, just try to get a better understanding how the system is connected. We looked at ecosystem coupling. This is not a very complicated thing. This is more or less just a correlation between everything that is there. And in our case, we said, let's look at community level. So we're moving away from the species level. We go to community and see how the communities are linked to one another when the animals are out, and see how these communities are connected to the abiotic environment. What we used was plant communities, above-ground invertebrates, soil microarthropods, soil nematodes, and soil microbes on the community side, and then carbon volcans and pH and moisture for trophics. Used the PCA for each community and took the first 2 axes just to have a bit more to kind of get the whole variability of that community. And what we have here, so these are the communities that may be plants, I can't remember, but maybe plant axis 1 and 2, and then this is the next community, the next, and so on. And these are the soil properties. This is the system, and what it is really just a correlation and the strengths of correlation between these individual data points. The fatter a line is, the stronger the correlation, and the bigger the circle, the more important that piece of the system is. As you can see here, we have quite strong connections or correlations between certain communities, but it doesn't really link that much to the abiotic environment. But when we take the mammals out, that changes, not so much the interaction, so we still have the systems still very well linked together, but it gets more linked to the environment. So the insects are able to hold up the interactions, more or less, and kind of the system is a lot tighter bound together. But when we kick the insects out, then everything falls apart and collapses, the interactions go out, and we lose quite a lot. So this was how it's connected. Then we thought, well, let's look at how this connects back to function of the system. These 6 functions is what we used to calculate the multifunctionality, just throw everything in the same pot. And if you look at ecosystem coupling here versus multifunctionality, not really anything. It's there's the trend, but not really. And then we kind of started taking it apart again. So here we have all the biotic interactions only, and there's no, the statistics disappeared, there's no significant relationship here. If we look at the biotic interaction, only we have a significant relationship between coupling and this is multifunctionality. So the more coupling we have in those interactions between the communities and the environment, the more important it is for the functions. Of course, we mostly use soil functions that could also be represented here. So, what's the big take-home message from this part? I guess what is important, I think, is the invertebrates are really important for the functioning of the system, and we really should not lose them. We should make sure we keep them around. The mammals didn't have, at least for the coupling, not that big an effect, but that doesn't mean that we shoot them all out and get rid of them. I think in our system we see that they're really important for the disturbing system to keep biodiversity of anything up. So whenever we had mammals, we had a lot higher biodiversity, and so it's really important to keep them too. That was the hardest point to argue with reviewers to write these papers. Okay, when you get rid of all the mammals, right? There's the problem, so you can shoot everything. No, we shouldn't. So that's the big take-home message, but could it be more complicated than what we saw here? So what happens, for example, if we have non-native species or invasive exotic species? We can have, for example, plants in the system. That's what we have here in a lot of cases. And you all know they're expanding a lot at the cost of the neighbors. You can hardly get rid of them, and they're economically a huge problem. But they're not just invasive grasses in a lot of parts, or exotics, but we also have domestic, which animals that come in the system here that may be not that big of a problem, or in Europe where a cow and the deer, cow is heavier than the deer, but functionally it may be a similar animal to interact with the system, but we don't really know that either, I think. And cows, you can see here by now, this is cattle, this is us here. Lion has nerves, this is cattle, there's some other fractions out here, but the most part here is cattle. This is the live mammals we have left. And most of them, again, these are the animals in the deer and so on. So yeah, cattle is a big thing, and then we also have places in the world, I take you down to Australia, where cattle actually is really exotic, because it's not a marsupial like all their animals, and it could be that in really not just the cows but also sheep and the rabbits, they probably have different function in these ecosystems. So what we tried is to repeat our experiment from home, exclude cattle first, then have the plot where only the native mammals and insects are, block out the native mammals, and then have again insect boxes that exclude the insects. We did that on farms, 6 different farms, where we have, in some cases, we have mostly native species, and some exotic, it's impossible to find a farm without invasive exotics. And then we had some farms that really look awful, only taken over by exotics. The exposure went up in November 2019. They look like this here. And you may see, it's not in yet, but Gregory here was interested in checking things out, so it seems that it worked. And the problem was 2019, we had big fires, so we were really lucky, we got scared and we didn't get burned down. And then came Corona, and then came Corona, and still Corona. And so finally this March, we were able to go around ourselves and start sampling. We have data, but I don't have any results. So stay tuned, that's a topic for another talk. I'm curious what we find and what we have in action as well. So, do we have all the players, or do we still have some parts missing here? For example, what happens if we have these large predators in the system that we don't have in our system in Switzerland? Could they play a role if they come in and influence everything else? For example, when they eat things as the wolf here is enjoying sludge in that zone. That's where I want to take you to Yellowstone National Park in the US, where I recently conducted a study on the effect of wolf kills on bison and rock-pitted deer, the rock-pitted deer and the bison, and what is the effect of when a wolf kills such an animal, what's the effect to the system. Looks like this. Here's the tonsil, so the content of the stomach that's left, and the couple bones, there's more bones here. And usually the wolf kill an animal, and they rip it open, so the gut contents go out, and all the blood and all the body fluids, they go to a place, and then they try to take the meat to some other place, because that part here stinks a lot. It still stinks after 3 months. We work with the wolf crew. They go, this is a winter kill here. The wolf crew GPSs that kills during winter from helicopters. Then they go later into measure bones and so on. And we got these data points to go in and see if we find the carcasses again. The reason is all the bears that are also in this system, so they did not allow us to go to anything that was younger than 2 or 3 months. So we're not going to walk in a bear and get in trouble ourselves. So then we looked at what happens to plant nutrient content and find it in the soil. We did that across the northern range, which is 120, 150 kilometers long, east to west, not so wide in north to south. But you can see here, these are it's hard to see here, but here's bison kill, elk kill. We had a total of 20 or 19 kills that we visited. We visited a lot more, but we learned with time that the small animal, the calf, is no longer to be found in spring. And some of the points were in water. We had snow in winter, and it was killed on the water, and by the time we got up the river, the carcass was swept away. We're also on high peaks, the carcass is just kind of blown downhill. A lot of hiking cases, a lot of cases for us was fun. So here's what we have again. This is that Hansen map. It's organic material that looks like hay, more or less. And so we measured on top of this, collected samples from here, and also from reference soil that looked more or less the same, or it was a slope, kind of tried to say the same elevational line, but going away from the impact that we have here. What we found was much higher activity of microbes, so CO2 emissions, under the course, under the carcass here. The plant quality was much higher for plants that grew in that area. That's all the bodily fluid that went into the soil, the blood, and the whatever content the animal has. But what we also found was a higher richness in the reference soil. So when you have a carcass richness drops, most likely there's specialized bacteria, fungi that go for all that stuff and decompose it. That was part of the result, but to me more exciting were the results of the community structure, what you have here. That's the reference soil here, and that's the carcass soil for fungi and bacteria. And you can see it changed a lot from the reference soil to the carcass. It was highly significant. We have completely different community. Same is true for bison, completely different. So that's what we have here. But what was also, and this was not expected at all, was this difference. That was significant. So we had a completely different fingerprint under bison carcass and under an elk carcass. And up to the point where we did that study, the only reference study we found were people laying out humans or pigs or mice somewhere in a barracking or so. And they always said there's no difference in the decomposing community of meat or flesh or decomposing mammal. It's always the same fingerprint. You're always going to find the same bacteria. Doesn't seem to be the case, and this could be for forensic science. That could matter as well, that know what species would have had which bacterial fingerprint to show. Yeah, so one of the take-home messages here that a large scale can increase or change the biodiversity at quite a small spatial scale. What we also found is a huge difference along the gradients we had. It was also said from these studies, there were some studies putting up as a human and mice. They always said soil doesn't matter, the soil composition or the soil, the texture doesn't matter, pH. It's always the same. There's no chance to find any differences in anything soil type-wise. Or it is, because we had quite a huge difference across the gradients. Also, the small north-south gradient we have was completely different of what happened with those communities. Yeah, so the pattern changed across the landscape, but you could say, okay, that's a carcass here and there. Who cares? They're not very frequent, and why should we worry about it? But if we look, this is a different system. This is Isle Royale National Park. It's Lake Superior, north of Canada. And the colleague of mine with whom I also did this study in Yellowstone, he worked at Royal for quite a long time, Joseph Tom, and he mapped he had data for 25 years of wolf-moose research in that case. And what he found is more or less in the end that after 25 years, the whole island, that's 25 kilometers, was touched at one point by a carcass. So with time that changes, and every spot in the system more or less get touched, so actually it matters that we have to change biodiversity in the soil in these systems. All right, so almost there, but we also have other animals in the system, and those are the ones that can disturb the system severely, especially in soil. You may be aware of the big compactors, the hippos, the elephants, they just dump things to density or higher density, but then we also have the ones that do the opposite. Biodegraders, as you have here, the arthropod in South Africa, the armadillo that moves a lot of soil, and we have our biggest one is the wild boar, but mostly for the ecosystems of the grasslands. But then we also have the smaller ones. Here, the groundhogs in the United States, they can have giant colonies in a lot of holes, so they dig a lot of soil up. And the same is also known for wolves. That's in our system. We have wolves that do the same thing. So they can have a big effect as well. But then we also have insects like termites that have more or less the same effect. And for this, I would like you to go with you to Africa and the Serengeti system, where we looked at the role of termite hills for soil functioning in processes. What we have here is a graph on soil texture, and we had gradients starting here, going 20 meters into the grassland. For soils, we only had a couple of points, and we didn't have the whole gradient. Normally, all the other points you're going to see is for the whole 20 meters out from the top of the mound. But what you can see here, that's the important part, is at the edge of the mound, we have a lot more clay than we have in the surrounding grasslands, and a lot less sand, so the opposite. But that means termites bring up the clay, and clay holds a lot more water, so there's a lot more grass growing in the dry season on these mounds, which is beneficial for all the ungulates that like to be around the mounds. What we also did, we looked at the CO2 evolution away from the mound soil CO2 emissions. Of course, you have here the high spot, that one, that's here. That's the respiration of the termites. That's what's coming out from through the funnels. But then, not really well visible, but we had a trend here towards higher emissions, then went down a bit, and we tried to see what's the reason for this. We tried it with soil temperature, small trend, no. Soil moisture, no. Vegetation height, or anything vegetation related, no. But the abundance of invertebrates in the soil away from the mound explained those emissions, and we found pretty large changes on that gradient. So the termites influence who else is living in the system and who's there. Again, hotspot, yeah, okay, maybe that doesn't matter, but if you look at the Serengeti ecosystem and termites mountain, yeah, it could matter because there's a lot of demands. Not in all parts of Serengeti, but a lot of areas look like this. There's termite mound after termite mound. And we tried to select mounds where people have gone into the next one, so the data should be unbiased from the next mound. And then, of course, something I don't really have much data for, but we have interactions with the ungulates. The ungulates like to go scratch themselves. There's completely different food around the mounds, completely different plants. They also use the mounds as lookouts to check around for predators and see if someone is there. And there's also, we looked at the seed bank here, there's different plants coming. Also in the dung, there's different plants for different animal species as well. And what we would like to do with our data eventually, but we just never really got to it yet, and the data is very difficult and heterogeneous and distant. A couple of times we gave up. But this will be to understand how these species interact through the dung, the seed bank, and then also the plants. But that's another stage, and hopefully we get a solution here as well. So bioinformation can matter. The impact of landscape distance elevation is huge. I guess also with those groundhogs in the US, it could be huge, and that's also been researched. But interactions to the higher trophic level or the large ungulates is not really understood. And also, what does a termite mound do with the predators? Is there an interaction also to the predator level? I think this is completely unknown today. Okay, last part, of course, biodiversity. Biodiversity loss, that's not the only thing. We also have nitrogen increases, climate change, and all these interact somehow. So what does that mean? I would have a lot of data, but that's the topic for a different talk. For example, look at where we have interactions between herbivore loss and nutrient emissions. So that should be something we should not forget. There's also other drivers in the system that matter a lot. So this gives us to the main conclusions. I think it's really important to see that both large and small are really important for an ecosystem. But I think still, if you go back to our airplane, I think we still do not understand which species are functionally redundant and which not. This is here a newer study by Stefan et al., an update from this diffusion function diversity. We don't know yet if we lost them already or not, or how many we can still afford to lose. So I think this is an important point to keep in mind. And I think it's, at least based on my studies, I think it's important to study an ecosystem quite comprehensively and to get as much data, because just individual parts that don't get old, like in my Swiss system, my total biomass composition doesn't change at all, but a lot of other things change dramatically. So I think to get a broad perspective of the system and also to get interdisciplinary research, I think is quite important. That brings me also to the last point, at least to me, I took it around the world. For me, it's important to see these systems and to get to know how they function in other parts of the world, because a lot of the literature that we have in science is from the global north, and it's completely different in cases in the south. And I think that's important to understand and do that interdisciplinary research. It's also fun, I think. I wouldn't be here if we wouldn't have met in Minneapolis 10 years ago or so. And I think it's important to see and get new ideas from different people and bring that into science and work together. So that's some of that international collaboration. Also a lot of people, which is usually also fun. And then that brings me to the last slide. There was no flag here yet. But we have a project funded by the Swiss National Science Foundation, Pedro, Laura, and Hugo Marrero is a Bahia Blanca. And what we will do is we look at the effects of livestock versus non and how that affects the ecosystem functioning and services, also biodiversity. And we'll do that cross-gradient from Brazil to through Uruguay, Argentina, along the Rio de la grasslands, starting in 2004, next year. For August 2004, we're looking for 2 students, PhD to come in the project. I'm excited to have this and we can get going. Then acknowledgements. I presented this research by far, it was not me. I had a lot of people, and I don't know, I'm an ecosystem ecologist probably, but if I do microbiology anyhow. But I enjoy working with different people and get to these borders of disciplines and understand the other part. To me, that's the fun part of research. So there's tons of people I worked with in the last couple of years, and without them, it would have been possible, a lot of these things. Then finally, thank you, and I'm sure you have questions, or I hope you have questions, and they can be in Spanish or English, just in Spanish. Thank you.
Questions, traen para charlar con Anita?
Just a philosophical question, really. When you say what we can afford to lose, what exactly do you mean?
What I mean is we're losing anyway. I mean, species are disappearing.
The way I see it, the environment as we didn't know it before Homo sapiens came into play. It's absolutely different, right?
Yes, we cannot go back there, but at the moment we are losing species, I think, at a faster rate than we did 50 or 100 years ago. And can we really afford to lose more, or could we? Maybe we can lose a deer, but maybe I don't know. We have to keep an ungulate in the system. That's nothing I did. We have like 3 ungulates, but can we afford to lose one, maybe the one with the lowest populations or not? Or can we afford to lose a cheetah in Africa to keep the lions? So I think we have to find out which are redundant, and if there's a redundant one, I don't think as an ecologist, I don't want to lose anything.
Sure, I agree.
Yeah.
It's just that
but there may be some that, if they're gone, somebody else can take the function. But I don't think we know who else will take over that function yet. Or if there's somebody left that can take the function, it's also very into that. Probably a more philosophical question now.
Because I'm sorry, I'm not an ecologist in any sort of way. I was just thinking that even if we make all animals disappear, this will stay working somehow, in some way.
No, never. I never. Because there's all these, you have the pollinators that need to do their job. Otherwise, if you don't have pollinators, we don't have plants reproducing. In the soil, we still don't understand anything, but there's certain species that make sure, the bacteria make sure that the nutrients get, decomposition process are formed, like in a compost, that things get turned over and the nutrients are available again for the plants. So if all these are gone,
What I meant in it was that in some point of historical times there were no animals on the land.
Yeah, but then
there was still some sort of
of course,
system.
yeah. But maybe I think if we lose everything, also we will be gone. But we could go back to bacteria or anaerobic things. I think that would work.
It would be boring, of course, but
yeah.
¿Alguna pregunta? I was also surprised about this idea of not changing at all biomass or soil composition, but all the big changes in the not the biomass or the total carbon and the result of the cell. There were no big changes in the structure of the soil system, but yes in the composition of bacteria and nematode richness and all that. But how do you think it would be possible to have these big changes in the structure of the soil community and not big changes in total carbon? Or
Well, I think the bacteria, the fungi, they did not change. Nothing really biomass, which is a lot of the carbon. I think that's why we didn't find any changes in the soil. Because all that bacteria didn't change and fungi didn't change. I think that's why we didn't find carbon and nitrogen changes. And I think that the nematodes that we have, they ate fungi, they ate bacteria, they ate roots. But it could be, one thing that could be that because we had an increase in the abundance of nematodes, that they actually ate the bacteria. And that's why we don't find a difference in the abundance of bacteria, biomass of bacteria. That's something we could not take apart. And that could also be why we don't have a change in soil carbon. Yeah.
Hi, Anita. Thank you very much. I would like to know if with these groups of nematodes or fungi that you have different functional groups. So if we can take that idea of Stefan of changes in functional diversity, if you have an idea of, for example, within nematodes, if there are more frugivorous or pathogens or herbivores, or within fungi, decomposer, pathogens, or mycorrhiza.
For the fungi, I don't know, we did not look into function. That was still done with your PFA and not genetic fingerprinting, the data we have. For the nematodes, we have functional groups. And I think in the paper that we published, there was we split everything up into the food web of the nematodes actually. I can't remember on top of my head what changed really.
But you can see those changes.
You can. It's pretty easy to, based on mouse parts, to see the different functional groups. Yeah, and there were changes. Yeah.
Okay. But you have not been studying fungi. There are more mycorrhizal fungi or decomposer or nematode or pathogens.
No, not at all. We just had really kind of a rough, not the fingerprinting, I guess, with PFA. The old PFA, we did not get to the.
Oh, PFA.
Yes. Yeah, but that's the old technique. Yeah.
And thank you for the nice talk. I like it. My question is, excluding big animals in general has opposite effect or different effect on the plant community. Sometimes it increases diversity or plant richness, sometimes it decreases. I think that now we know that maybe it depends on the plant dominance or the dominance of the plant community. Do you think there are some generalization related with the effect on insect diversity or the other microbial communities diversity? After looking at all these,
I think there's really not a lot of research on invertebrate plant. I mean, there's a lot of invertebrate plant interaction, but that's a lot of pot experiments or small mesocosms in the greenhouse. But ecosystem level-wise, there's a huge gap, and it's hard to get them separated and out. And we had control systems, so we had also had cages to make the same light conditions and moisture conditions to see if actually it's the insects or if it's the light and the UV changes that we have to find that biodiversity. There's other people that spray to get rid of the invertebrates, but I think there's really very few experiments. Now with the bugnet, that's a new international experiment that is set up. They do spray insects across the globe, I think, on plots. I think with that we should get a better idea of what's going on. And with the soil, also especially soil invertebrate, that's still the big black box of we don't know in the soil.
¿Una otra pregunta? Bueno, muchas gracias. Sí, Martin.
Well, Anita, how do you compare your results in which you remove natural mammals with the literature that compares the effect of removal of domestic animals? So the question is, to what extent we as humans sort of replace mammals, natural mammals with domestic animals, and some of the ecosystem functions have been kept?
There's I've been just reviewing for an article I'm writing that literature. It seems that there's at least the evidence is that wild animals function quite differently from domestic animals, or the effect on biodiversity and also the paper I went through was on biodiversity and also soil and carbon storage. There's a different effect of domesticated animals and wild animals. And I think, if you replace them, we probably get the interactions keep going, but there's a different footprint we will have. At least we have something that eats stuff. Like here, I think for also our new project, it's not in the northern part of the world, we always say, or that's a new thing, they say, okay, take the cattle out of South America so we can save the climate, which is economically not a good idea, I think, and ecologically not. Looking at the Las Chicas with the fescue that comes in and dominates everything, I think this is not the solution. So I think at least in some parts of the world we have an animal that's grazing and continuing a function that should be taken care of. But it's a different role they have.
Sorry. Hey, Erika. I have a question regarding climate. So you compared very different sites, right? Cold ones in Switzerland, Tanzania, etc. And I was wondering if the effect of the removal of big or large mammals was more marked, I don't know, the colder biomes or in the hotter ones.
Well, these are what I presented are all case studies, so it was always something like different experiments at different places. I see, based on the NUCNet data, we see a pattern, but it changes. So the context of where you remove the animals the response is context dependent. It's aridity, it's temperature, it's rainfall, but also the soil that you have, nutrient levels in the soil, and so forth.
But also biodiversity is different, right? So
yes, yeah. But here you cannot really make comparisons, these are just like individual case studies.
Bueno, muchas gracias. Don't know the lunch, but if you want to talk, she will be here for the next 2 weeks. Here 1 week, and then Bangladesh. So, thank you, and see you next Friday.